Hi everyone, welcome to IGCSE Study Buddy, where you can revise biology topics from the Cambridge IGCSE syllabus. If you are enjoying these videos so far, please don't forget to hit the like button and subscribe to our channel. In this video, you are going to learn part 2 of chapter 17, Inheritance. Before we begin this section, there are some important definitions you must learn. So we already learned that inheritance is the transmission of genetic information from generation to generation. A gene is a section of DNA that provides instructions for making a specific protein which influences a particular characteristic. Alleles are variations of the same gene. Since we possess 2 copies of each chromosome, we also have 2 copies of each gene, resulting in 2 alleles for each gene. These alleles come from our mother and father, which means they may not carry the same information. For instance, when considering the gene responsible for eye color, an individual could have one allele for brown eyes and another allele for blue eyes. Genotype refers to the genetic makeup of an organism, including all of its genes and alleles. It represents the combination of inherited genes that determine an organism's characteristics. Phenotype is the observable features of an organism. So it can be a feature that we can see, like eye or hair color, or it can be a feature that we can find, like blood type. Homozygous is having 2 identical alleles of a particular gene. 2 identical homozygous individuals that breed together will be pure breeding. Heterozygous is having 2 different alleles of a particular gene. A heterozygous individual will not be pure breeding. So for example, say we are talking about a gene with the alleles capital A and simple a, the homozygous genotypes will be either capital A or simple A, simple a. So homozygous means having 2 identical alleles. The heterozygous genotype will be capital A, simple a. Heterozygous means having 2 different alleles. We normally write the capital letter or the dominant allele first. Alleles can be dominant or recessive. A dominant allele is an allele that is expressed if it is present in the genotype. A dominant allele only requires one parent to pass it on for the trait to be visible in the physical appearance, that is the phenotype. A recessive allele is an allele that is only expressed when there is no dominant allele of the gene present in the genotype. So, a recessive allele needs to be inherited from both parents for the trait to be visible in the phenotype. If there is only one recessive allele present, it will remain hidden and the dominant trait will be expressed. Dominant alleles are represented by capital letters, while recessive alleles are represented by the same letter in lowercase. Monohybrid inheritance is the study of how a single trait or characteristic is passed from parents to their offspring. A Punnett square is a helpful tool to determine the possible combinations of alleles in offspring. It allows us to calculate the ratio of these combinations. Remember, in a Punnett square, dominant alleles are represented by capital letters, while recessive alleles are represented by the same letter in lowercase. For example, the human eye color is controlled by a gene that has alleles. One allele for this gene may code for brown eyes, whereas another allele may code for blue eyes. Say the brown eye allele is dominant and is shown as capital B. The blue eye allele is recessive and is shown as simple B. If a question doesn't specify the letters for alleles, you can choose any letter to represent them. Just make sure to use the same letters consistently in your discussion and avoid using letters that look similar in their capital and lowercase forms. For example, like capital C and simple C. So the homozygous dominant genotype for brown eyes will be capital B, capital B. And the homozygous recessive genotype for blue eyes will be simple B, simple B. In the first example, let's cross a pure breeding brown-eyed parent that is homozygous dominant capital B, capital B with a pure breeding blue-eyed parent or homozygous recessive simple B, simple B. So remember, the term pure breeding indicates that the individual is homozygous for that characteristic. To make a Punnett square, put one parent's alleles on the top row and the other parent's alleles on the left column. The cells in the square will show the different combinations of alleles from each parent. The results you get in the square will be the offspring, the babies. So the genotype of the pure breeding brown-eyed parent will be capital B, capital B. And the genotype of the pure breeding blue-eyed parent will be simple B, simple B. The gametes will be capital B and capital B for the pure breeding brown-eyed parent. And for the pure breeding blue-eyed parent, it will be simple B and simple B. So now we cross them. Each cell represents a potential offspring's genotype. Please make sure to write the dominant letters first. The Punnett square will look like this. Here we will have capital B, simple B. So the offspring will have brown eyes, but this genotype is heterozygous. Here we will have capital B, simple B as well. Here we will have capital B, simple B. And also here we will have capital B, simple B. By completing a Punnett square, you can predict the probability of various outcomes in monohybrid crosses. So as you can see, there is a 100% chance that all the offspring will have brown eyes. When it comes to eye color, having just one copy of the brown eye allele, capital B, is enough to show brown eyes because it is dominant over the blue eye allele, simple B. So if a parent with pure brown eyes, capital B, is crossed with a parent with pure blue eyes, simple B, all the children will inherit one copy of each allele, capital B, simple B, and they will have brown eyes like the brown eye parent. Next, let's say two heterozygous parents are crossed. What are the possible allele combinations of the offspring produced? So the genotype of one heterozygous brown-eyed parent will be capital B, simple B. And the genotype of the other heterozygous brown-eyed parent will also be capital B, simple B. The gametes will be capital B and simple B for both the heterozygous brown-eyed parents. So now we cross them. The Punnett square will look like this. Here we have capital B, capital B. So this will be a pure breeding brown-eyed offspring. Here we will have capital B, simple B, a heterozygous brown-eyed offspring. Here also we will have capital B, simple B. And here we will have simple B, a homozygous blue-eyed offspring. So in this cross, there is a 3:1 ratio of brown eyes is to blue eyes, meaning each offspring has a 75% chance of having brown eyes and a 25% chance of having blue eyes. In the next example, let's say a heterozygous parent is crossed with a blue-eyed parent. That's a homozygous recessive. So the genotype of the heterozygous brown-eyed parent will be capital B, simple B. And the genotype of the blue-eyed parent will be simple B, simple B. The gametes will be capital B and simple B for the heterozygous brown-eyed parent. And for the pure breeding blue-eyed parent, it will be simple B and simple B. So what are the possible allele combinations of the offspring produced? Here we will have capital B, simple B, so the offspring will have brown eyes. Here also we will have capital B, simple B. Here we will have simple B, so this offspring will have blue eyes. And here once again we will have simple B, In this cross, there is a 1:1 ratio of brown to blue eyes, meaning a 50% chance of the offspring having brown eyes and a 50% chance of the offspring having blue eyes. Now let's learn how to use a test cross to identify an unknown genotype. So for example, let's say we are studying a person with brown eyes of unknown genotype. How can we determine whether the person is homozygous dominant, that is capital B, or heterozygous, that is capital B, simple B, for the brown eye color trait? So what we know is that the brown-eyed person may be either homozygous dominant, that is have the genotype capital B, or heterozygous, that is capital B, simple B. To determine the genotype of an individual with an unknown eye color, you can cross them with someone who has blue eyes, which represents the recessive phenotype. So the genotype of the homozygous brown-eyed parent will be capital B, and the genotype of the blue-eyed parent will be simple B. The gametes will be capital B and capital B for the homozygous brown-eyed parent, and for the pure breeding blue-eyed parent, it will be simple B and simple B. These are the possible allele combinations of the offspring produced. Here we will have capital B, simple B, so the offspring will have brown eyes. Here also we will have capital B, simple B. Once again, capital B, simple B. And here as well, capital B, simple B. So as you can see, there is a 100% chance that all the offspring will have brown eyes. So this confirms that the person is homozygous dominant, that is capital B, as they are only passing on the dominant allele, capital B, to their offspring. On the other hand, let's assume that the brown-eyed parent is heterozygous for this gene. So the genotype of the heterozygous brown-eyed parent will be capital B, simple B. And the genotype of the blue-eyed parent will be simple B. The gametes will be capital B and simple B for the heterozygous brown-eyed parent. And for the pure breeding blue-eyed parent, it will be simple B and simple B. These are the possible allele combinations of the offspring produced. Here we will have capital B, simple B, so the offspring will have brown eyes. Here also we will have capital B, simple B. Here we will have simple B, so this offspring will have blue eyes. And here once again we will have simple B, In this cross, there is a 1:1 ratio of brown to blue eyes, meaning a 50% chance of the offspring having brown eyes and a 50% chance of the offspring having blue eyes. If any of the offspring have blue eyes, it would confirm that the person is heterozygous, that is capital B, simple B, because the presence of the recessive allele, simple B, would result in the expression of the blue eye color. You have to be able to understand pedigree diagrams for the inheritance of a given characteristic. Pedigree diagrams are used to track how a specific trait or disease is passed through generations in a family. They can help predict the chances of inheriting the genetic disorder. In a pedigree diagram, males are shown as square shapes, while females are shown as circles. Affected individuals are shown in red, while unaffected ones are shown in blue. Horizontal lines connecting males and females show that they have offspring, which are displayed beneath each couple. Codominance is a situation in which both alleles in heterozygous organisms contribute to the phenotype. Inheritance of blood group is an example of codominance. In the inheritance of blood groups, there are 3 alleles for the gene IA, IB, and IO. These 3 possible alleles can give us the following genotypes and phenotypes. The alleles IA and IB for the blood group gene are codominant, meaning that they both show their effects when present together. They are both dominant over the IO allele. The gene is represented by the letter I, and the superscripts A, B, and O represent the different alleles. Let's look at a genetic diagram to predict the outcome of a cross that involves codominant alleles. So for example, say we are asked to show how a parent with blood group A and a parent with blood group B can produce offspring with blood group O. So the genotype for the parent with blood group A will be IAIO. And for the parent with blood group B, it will be IBIO. The gametes will be IA and IO for one parent and IB and IO for the other. We know that these are only their genotypes and not IA or IB because according to the example, we know that they are able to produce a child with blood group O. And so the child must have inherited an allele for group O from each parent. Also remember to write the genotype with the dominant letters first. So now we cross them. Here we will have IAIB, which is an offspring with the blood group AB. Here we will have IAIO, which is blood group A. Here we will have IBIO, which is blood group B. And here we will have IOIO, which is an offspring with the blood group O. So as you can see, there's a 1 in 4 or 25% chance of a child with blood group O. There's also a 25% chance of producing a child with blood group AB, a 25% chance of producing a child with blood group A, and a 25% chance of producing a child with blood group B. Finally, let's learn about sex linkage. A sex-linked characteristic is a feature in which the gene responsible is located on a sex chromosome, and this makes the characteristic more common in one sex than in the other. Red-green color blindness is an example of sex linkage. It is a genetic condition that is caused by a mutation in the genes located on the X chromosome. Let's use a genetic diagram to predict the results of a cross involving sex linkage. In this example, we'll consider a cross between a normal male and a carrier female, where X capital R represents the normal allele and X simple R represents the color-blind allele. So the genotype for the normal father will be X capital RY, and for the carrier mother it will be X capital R, X simple R. The gametes will be X capital R and Y for the father, and for the mother it will be X capital R and X simple R. So in the carrier mother's genotype, the dominant allele masks the effect of the recessive allele. So even though she doesn't have the disease, she has a 50% chance of passing it on to her offspring. Please make sure to put X chromosomes before Y, and you also write any sex chromosomes with the dominant letters first. So now we cross them. The Punnett square will look like this. Here we will have X capital R, which is a normal female. Here we will have X capital R, X simple R, which is a carrier female. Female because XX, right? Here we will have X capital RY, which is a normal male. And here we will have X simple RY, which is a color-blind male. So as you can see, there is a 1 in 4 or 25% chance of a male child with color blindness. There's also a 25% chance of producing a normal female, a 25% chance of producing a carrier female, and a 25% chance of producing a normal male. The inheritance of red-green color blindness is more commonly seen in males, as they only have one X chromosome. Females, on the other hand, need to inherit 2 copies of the color-blind allele, that is X simple R, X simple R, to exhibit color blindness, as the normal dominant allele X capital R on their other X chromosome masks the effect of the recessive allele. So that concludes Chapter 17, Inheritance. Hope this video helped you. Please share your thoughts and suggestions in the comments section. Thank you for watching, and please don't forget to subscribe to IGCSE Study Buddy for more biology revision videos. Bye-bye.